In the meantime, if there's any doubt, we can take that up. And so today's class will settle our debate around the CD40 and CD40L molecule. So let's look forward to it. Okay, good evening to both of you. So I think let's just proceed with the class, and if others have to join, they can join in the meantime. So for today's class, the topic is it's a Nobel Prize winning concept, so you can expect it to be slightly heavier than what we have been discussing so far, and also take this class as an opportunity for you to keep on putting everything together in context, right? Because we have been discussing quite a few things, quite a few details we have already discussed. And just to bring everything together, this is one, we will now gradually be discussing topics that will help you assimilate everything together in the same picture. It is very important that all of you also try and visualize what is exactly happening. Otherwise, it's a lot of details, and we might just get lost in the entire subject trying to understand what exactly is happening. So, if we like always try to bring things together, and that is going to really help you in overall understanding of the immunology concepts. Okay, so let me just present my screen.
Recording in progress.
So, just to quickly have an overview of what we were trying to discuss in the previous class. So, the last three classes are a bit related to each other, including today's class. So you will get the notes for all these three classes together. If so, just as we get to complete this topic of B cell development, maturation, all the receptors, how the diversity in the receptor is generated. Once we finish up with this entire topic, you will get this entire handout together for your references. So if you will not, you might have not found this section already in your handout sections. So that's the reason behind it. So this is our the first diagram that we started our discussion with while we were discussing about the process of maturation activation of B and T cells. So far we have been discussing about B cells, and I think another class of discussing about the B cell responses, the antibody responses also we are counting for that in the B cell responses, and then we will move on to our T cells. So we saw that in the process of development, there is an antigen independent phase that is essentially happening in the bone marrow. And once the naive cells come into the peripheral organs, peripheral lymphoid organs, we have the beginning of antigen dependent phase where the binding, so over here, immunocompetency and antigenic commitment was achieved. And once this comes into the circulation, if it finds an antigen of appropriate make, it gets to bind to it and it initiates a sequence of events, which we have all discussed. Now, slowly, so clonal selection we have discussed very well. We will see that why this IgM and IgD. So we discussed that there's IgM and IgD molecule that forms onto the surface of a naive B cell. We'll look into the fact that how only these two transcripts are produced. and how when there is class switching, how the this switch is exactly happening to the other antibody types. The different antibody types that we also try to just briefly take up because those are very straightforward concepts. And if you have any doubts from there, that you can get back to me with. So this is a basic structure of antibody that we have discussed, the basic types of it. They have respective some distinct function for each of them. We just quickly discussed all of it. And now we will move on to understanding that how whatever we have read so far is actually taking place, right? So today's class is going to answer your hows in the subjects that we have discussed so far. So again, uh, we'll begin it with addition of certain details, so a little more detail into what we have already learned. We read that about the antigen dependent and independent phase. In the antigen dependent phase, let's just first come here. So, lymph node is one of the most important lymphoid structure where the entire process of antigen encounter is happening. So, your antigen encounter is triggering the effector response is essentially coming up once your antigen encounter is happening. So, what happens that we have a peripheral lymphoid organ. So, here we have taken the example of lymph node. Inside the lymph node, we have distinct anatomical sites. So we have uh, we discussed that there are certain regions. Again, that is right from our third class. So you have to, that's what I'm saying. You have to like try and recall everything that we have already discussed. We have discussed that there is something called as cortex, which is the outermost side of the organ. Then there is paracortex and the medulla region, from where we have the efferent lymphatic vessels taking out whatever has been produced inside, whatever molecules have to come out into the lymphatic system. This efferent lymphatic vessels carries it out of the lymph node. And we have the afferent lymphatic vessels, which brings in the antigen along with the lymphocytes, right? So inside this lymph node, and so that's the case also with all the peripheral lymphoid organs. We are just taking in detail the lymph node structure because that is one of the most important peripheral lymphoid organ, firstly. And all the different mechanism that you actually see happening here, you will also find that in your uh, say the spleen that we have also discussed. So we saw that there is something called as primary follicle, which then forms a germinal center, and ultimately this follicle with a germinal center is called as a secondary follicle, right? In the cortex region, we have these primary follicles developing. We have now read about B cell activation. So where is that B cell activation happening? It is happening in this cortical region where a primary follicle is present. As soon as this primary follicle encounters an antigen, it transitions into a secondary follicle. Which antigen? We have discussed the thymus dependent and thymus independent antigen. So whenever we have an antigen coming up, within 7 to 10 days of its exposure, we have these secondary follicles forming what we call as a germinal center. This germinal center is nothing. So inside this secondary follicle you see here, the right core of the center of this germinal of the secondary follicle is designated as a germinal center. This is the region of the most active, this is the most active site for B cell proliferation. These proliferating these proliferating B cells are called as the centroblasts. Just a second. So blast is essentially something that divides. Anything that has the capability of dividing, we provide it with the suffix blast, right? So inside this germinal center, we have the B cell proliferation happening. So these proliferating cells are called as centroblast, and they ultimately differentiate into plasma cells and memory cells. We have understood this so far. We will see that exactly what are the steps that comes in this process of differentiation over here, just in a couple of minutes. Once we have these two cells forming, the plasma cells and the memory cells, the third process is from this. So this primary follicle was there in the cortical region. The secondary follicle is somewhere around the margin of the cortex and paracortex. The paracortex is where the T cell and B cell encounter is happening. In this process, both T and B cells get activated. Why so? Because over here, the B cell that gets activated, it is having the ability to express class 2 MHC molecule. So whenever there is a class 2 MHC molecule, the cells have the ability to present the antigens to the CD4 plus T helper cells. So the CD4 plus T helper cells in the margins of this paracortex are now getting activated because the antigen is being presented to it by the activated B cell. The T helper cells in return is going to communicate back to the B cells where the B cell activation is further augmented and it gets a chance to further differentiate into the plasma cells and the memory cells. So that's why paracortex is a site for initial T and B cell activation. Once we have these products, the plasma cells and the memory cells, we need these plasma cells to come out into the circulation, and that's why they reach the medullary region. From this medullary region, they get a chance to drain out in the efferent lymphatic vessels, right? So these are the three steps that is essentially happening inside the lymph node. But if you would notice, the most important region of this is the germinal center, because inside the germinal center, the entire process of B cell division is happening. And this complicated looking diagram explains how the entire process is happening. But once we break it down, I hope this entire diagram feels a little more simplified for you all, right? So the core part, the most inner part of this germinal center is known as the dark zone. And this is the place where somatic mutations are happening. For now, just remember this as a term that has come up. So somatic mutation, if you try and break it down, and also if you have gone through the recorded lectures of the faculty, you must have encountered it already, that somatic mutation is bringing up the diversity in the make of antibody, which enables it to interact with a variety of antigen present in the environment. So how this is happening, that is what we will discuss towards the end of today's class. But for now, we have B cell population, which has an affinity. So affinity can be quantified in terms of certain numbers. So it's a quantifiable entity. They have just denoted that the affinity is K1, right? So this B cell population in the primary follicle, or maybe coming in from this afferent lymphatic vessel, they get in and they now get a chance to enter the germinal center. Inside this germinal center, the activated B cells or the centroblasts, how do we identify them? They have comparatively large size, the chromatin is more diffused, and we do not see any surface immunoglobulin molecules on the centroblasts, right? So this centroblast enters the dark zone of the germinal center. Because it do not have any Ig molecules, so a lot of mutation scope is present. So the cells are supposed to go under somatic mutation. By this process, when they divide, they are going to give rise to cells which are now called as centrocytes. Centrocytes have now become non-dividing. The first thing, the centroblasts divide into centrocytes, and the centrocytes will not divide further. In this process, where the centroblast division into centrocytes was happening, two things are happening. One is the somatic hypermutation that we are saying. And as a consequence, why is this somatic hypermutation happening? It is happening to bring about what is called as affinity maturation, which means that if the affinity of a B cell population that entered the germinal center was K1, let's just say, and the affinity here and the affinity here, say it becomes K2. So in the process of affinity maturation, what happens that this K2 is much, much greater than K1. So the increase in this affinity is actually happening because of the process of somatic hypermutation. So while we were trying to discuss about the antigen antibody interaction, where we did not take it up in a lot of detail, but we did see that the clonal selection process is happening. How is it happening? That we already have an antigenic commitment. So an antigenically committed antibody molecule gets to bind this antigen, and we said that there is nothing like a induced fit model. So this antigen is not going to make any major change in the binding ability. So it will not cause the antibody to bind to it, irrespective of the fact that whether or not initially it was capable of binding it. That doesn't happen. But once this antigen gets to bind this antibody, some interaction happens that is, so this process we called as activation. In this process of activation, the antibody molecule is going to undergo certain mutation. Because of these mutations, so just one or two base pair mutation is going to be caused. Because of these mutations, this binding affinity is going to be further higher compared to what was initially there. So this process is known as affinity maturation, and it helps to stabilize the overall antigen antibody interaction that is already happening. Now, apart from the this positive selection effect, we can also have, because these mutations that are happening, these are essentially just random mutations. The body is just trying to produce as many mutations as possible. Somewhere near the rate of 10 to the power minus 3 mutation per base pair, which is, so you might say that there's just 10 to the power minus 3, but if you remember from, say, if you have read molecular biology, you would know that the chances of mutation coming up is somewhere in the order of 10 to the power minus 9. So compared to that, it is almost a million times higher chances of mutation that you see when we are looking into the immune cell development, right? As a consequence of this random mutation, we can have a centrocyte which now has a higher affinity, but we can also have a centrocyte which has lower affinity. The body do not want something that is regressive in its production. So why would it want to keep something which do not have any beneficial effect compared to the initial centroblast? So such cells are taken for apoptosis. Apoptosis is a, I'm sure you all have come across this word. So apoptosis is what we also break it down as programmed cell death. So if you just break it down, programs, there's a proper pathway by which the cell is ultimately triggered for death. So anything that is not useful to the body, it asks the cell to undergo apoptosis. The body has enough signaling mechanisms in place which ensures that every non-useful cell is triggered for apoptosis. So this high affinity centrocyte, the hero cell that has now come up after a lot of division. During the division, it has accumulated a number of hypermutations, and now this can bind to the antigen which is presented to it by this octopus-like structure that you see. This is called as the follicular dendritic cell. So follicular dendritic cell, we have also encountered the name of this particular cell type while we were discussing our lymphoid organs. Again, if you do not recall anything, please go back to the previous classes, and you will have to always try and bring things together whenever you are studying about immunology, right? So follicular dendritic cells, which are stationed inside the lymphoid organ, they have the ability to bind, to express the antigen onto its surface. So these small, small red dots that you see over here, these are the antigens that are expressed by the follicular dendritic cells in the light zone of the germinal center. So, this, let's just say that this is the entire secondary follicle. The innermost core region is the dark zone. More towards the peripheral region is the light zone. And from the light zone, the cells now come out of the follicular make that it assumes, and they get a chance to drain into the efferent lymphatic vessel, right? So inside of this this light zone is the area where we have our follicular dendritic cells stationed. They select for these high affinity centrocytes. How? So these antigens that are stationed on the surface of the follicular dendritic cells, the antibody molecule which is present on the surface of these centrocytes bind to these antigen molecule. This binding is essentially so let's just say that this is a magnet, and everything that has a strong affinity, every iron nail that has a stronger affinity for binding to the magnet gets stick to it, and the aluminum pieces which are of lower affinity, they are triggered for apoptosis. So just like it's like elaborate processes are there where antigen is presented on its surface. Whichever centrocyte binds to the antigen that is being presented, they get the chance to stick to this follicular dendritic cell, and that is how the selection for this high affinity centrocytes is happening. If at any point any one of you feel lost, please stop me right there, and we can first clear our concepts from the very beginning rather than trying to hold it until the end of the class, right? Okay. So this centrocyte that binds to follicular dendritic cell is now going to get a chance to interact with the T helper cell. One thing, so first signal that happened is the binding of the centrocyte to the antigen. Important fact that we must remember is that these follicular dendritic cells, they are different from the dendritic cells that come from the say the bone marrow part of it. So this has come from the lymphoid origin. If I am not wrong, just revisit the class. So we had two origin, the myeloid origin and the lymphoid origin. We had dendritic cells that were coming from both the lineages, right? So the follicular dendritic cells here, they do not have class 2 MHC molecule. So they cannot, so they are not the antigen presenting cells. Antigen presenting cells present antigen to CD4 plus T helper cells. Here, the antigen is being bound by the receptors present on a B cell, by the antibodies present on B cell, not to the CD4 plus T helper cells. So follicular dendritic cells are not antigen presenting cells. They only help to retain the complex formation that happened between the antibody molecule of centrocyte and the antigen that is present on the surface of this follicular dendritic cell. So you can imagine it like a mesh which traps, maybe like a filter that gets to the trap cells with a particular selectivity, or you can just imagine it like a magnet that is stationed to trap cells which have a desired affinity, right? And to ensure this trapping, it needs to try and maintain this interaction as far as possible. So the dendritic cells essentially have the function of only presenting antigens to the developing centrocytes, and it tries and stabilizes this antigen antibody complex that is formed. So once this binding of antigen happens, we have the selected centrocyte now getting a chance to interact with the T helper cells, which is also present in the paracortex region, right? So for this interaction, the most important interaction that happens to maintain the activation, maintain the selected state of the centrocyte is the CD40 on the B cells and the CD40L molecule on the T helper cell. This interaction between the B cell and the T helper cell is going to uh, give a positive signal to this selected centrocyte. along with it, but now the B cell here is an antigen presenting cell, so it has MHC2 molecule. The antigen that was presented to it by the follicular dendritic cell, this B cell, the selected centrocyte, has internalized, say, that antigen and now is presenting that antigen with the class 2 MHC molecule present on its surface. This presentation, the antigen MHC2 presentation to the TCRs, the T cell receptor present on the T helper cell. So there are three such interaction that is actually activating your centrocyte to form now then next differentiate and then produce the effector molecules into the lymph, right? What are those three interactions? First, binding of antigen, the CD40 and CD40L interaction between B cell and T helper cell, the antigen MHC complex interaction with the TCRs. These three interactions together ensures the activation for your B cell. In addition to it, class switching, a phenomena that we will learn about just for your reference right now. So we saw that the initial naive B cell has IgM and IgD molecule bound to its surface. But while we read about the types of antibody molecules, there are 5 antibody molecules. So of the 5, how that switch is happening, so the IgM molecule is going to change into which isotype of the immunoglobulin, say IgG or IgA. That switch is also determined by this interaction of the CD40 and CD40L molecule, right? So now, once we have all the interaction that the B cell had to undergo for it to get selected, it now gets a chance to give its effector, it get differentiated into plasma cell, or first the plasma blast, which will become a plasma cell that can either produce antibodies and release the produced antibodies are released into the lymph that takes care of the antigen, or this plasma cell can also come out of the germinal center in itself and gets a chance to reach the circulatory lymph or the blood, wherever it is required. The memory cell here, this also has the same function. So it can either be present in the inside of this lymph node or it gets a chance to move out, but its job is to just retain the memory of whatever antigen encounter has happened, right? So this light zone is also the zone where class switching, maturation happens. Class switching, the process of maturation and memory essentially only happens in the germinal center. So if for say any sort of deficiency, the body do not have the ability to organize a secondary follicle with a germinal center, the process of maturation and memory formation is not going to happen. The formation of plasma cells still might be there, and this class switching mechanism can also happen outside of the germinal center. But maturation and memory formation can only happen inside of a germinal center. So just take a minute to have a look on this entire figure. Give me a minute's time. I'll be back, right? क्या बोली जानी Do we have any doubts on this? Anything, anyone? There's usually two conditions where we do not have any doubt. When we understand everything or when we don't understand anything. So I hope it's the first case with all of you. Okay. So this diagram is essentially adding of the details to what we have already discussed, and we have also got a chance to see some of the phenomena that is happening, and we'll see how that is happening in the course of our following discussion, right? Now, so we have been talking so far about, so we discussed that the antigenic diversity that is there is huge, right? So in the entire environment that you're surrounded by, you have so many pathogens, so many so many different types of molecules present on these pathogens, and all of which is being recognized by your B cell receptor or T cell receptor with tremendous specificity. So specificity is a component of the cell receptors that we have discussed from the very first class, right? Today's class is actually going to clarify to you that how the specificity component, the diversity component is achieved by the immune cells. So we also discussed that the order of diversity that is produced for antibodies, the number of antigens or the different types of antigens that it can encounter is somewhere in the order of 10 to the power 10 specificities that the antibodies that the body can produce antibodies against, right? How is that achieved? That remained a question in immunology for quite some time. The main so the main problem that the scientists were facing is the germline DNA is not having so many genes that can actually encode all the diversity that happens, right? So the germline DNA, when people try to sequence the protein and then sequence back the genes from which these proteins could be coded, the number of genes is huge, right? So that was coming somewhere beyond the entire number of genes which is present in the humans. So that was a big question, that then what is coding this diversity? The other possibility that came is that there is somatic mutation. Somatic mutation that when we have say a mRNA transcript has formed, or maybe in the periphery when a gene is present, it is undergoing a lot of mutation. It is accumulating mutation as a result of improper DNA replication, or maybe different splicing coming together, splices coming together, which is resulting into this huge diversity that we see. But either models independently could not explain the range of diversity that was actually coming up. Because even if it's a faulty replication happening, how faulty and how come RNA polymerase or DNA polymerase is suddenly causing that many errors to particular gene types only, right? So with all these confusion around, two concepts came up which to the best try to explain the mechanism, try to reason the this phenomena that was being encountered. One is a multi is the multi-gene organization of the immunoglobulin gene. The other being that the immunoglobulin genes get a chance to rearrange. Both of them in itself was a big statement to make, because one gene, one protein hypothesis was the most prevalent, most accepted hypothesis at that time. So one gene, one protein hypothesis says that if you have one protein forming, it must be forming, if we backtrace it, it must be forming from a particular gene sequence encoding it. But over here, we saw that now since we know that there is two chains in the antibody molecule that we discussed, the heavy chain and the light chain, and both this heavy and light chain is actually encoded by different uh, gene segments. That part was still straightforward to understand, that we have two different polypeptides, and we have certain linkages that might be linking the two together into a, so we have two proteins, and we have certain linkages that links the two proteins together into a polypeptide, and this was still reasonable and understandable, right? What was more difficult to understand is that this one protein, instead of being coded by one gene, we have multiple gene that is coding this protein. Similarly, multiple gene that codes for this entire structure. So people came across the fact that for the heavy chain, we have four gene segments that is encoding one protein. And for light chain, we have three such gene segments encoding the light chain. So this C essentially codes for the constant region that we have talked about. So we talked that in the light chain, there is a variable region and there is a constant region. Similarly, for the heavy chain also, we have a variable and constant region, right? So this C gene segment would always code for the constant region in either chains, right? So it's still it's still on different chromosome, the same gene segment, but for both the proteins, we have a C gene segment somewhere near the vicinity of these gene segments, and they are in some way, they are going to come together. We'll just see how that happens. V, as you might guess, it's for the variable portion of your heavy as well as the light chain. And D and J segments are in addition somewhere in between the V and C segments. Each V gene segment is preceded at the 5 end by a small stretch of nucleotides, which is named as the signal sequence or the leader sequence. We'll just try to visualize everything in the following diagram, but just for a fact, you must know. So what we can understand from all the things written over here is that there must be a gene present like this, which can be a V gene. So this is one chromosome. So entire chromosome can have multiple genes arranged on it, right? So we have a V gene, we can have a D gene, J, and C. Similarly, for the light chain, this sort of possibility is there, right? So some linear sort of arrangement is what you can visualize from whatever is written right now. Apart from the fact that this being a single stretch is one thing, and then came the concept. I should not have did it. Then came the concept that these genes can rearrange. This can join here, this can join here. All sort of combinations can be formed because of certain sort of rearrangement that is possible for immunoglobulin genes, right? Okay. Now from your previous class, you would remember that the variable chain so the antibody molecule that forms always have either the variable chain can either be kappa or lambda, right? So there are two possibilities, but it will never be like one chain is kappa here and the other chain is lambda here. That is not, that is never going to be possible, right? And we also read about that these heavy chain can also have 5 isotypes based on whatever is the domain and everything present on it. So we will, since So one thing is types of heavy chain, which determines the isotype, and the other fact is the possibility of a kappa chain or a lambda chain. So these are completely so two different genes can actually it's completely on a different chromosome, and we have separate genes on separate chromosome that either of them get a chance to get produced and get assembled with a heavy chain to form an antibody molecule. But your heavy chain, on the contrary, all the different isotypes, constant regions of all these different isotypes are all arranged in on the same chromosome. Let's just assume like it's one by one, either IgG corresponding constant region is present or IgD corresponding constant region is present. All such constant regions are arranged on the same chromosome. Here, they are completely on different chromosomes, right? So, just a comparison that you can have the genes corresponding that make up the lambda light chain. In humans, it is chromosome number 22. In mouse, it's 16, kappa 2 and 6. Heavy chain for all, but all the different isotypes, based on the different heavy chain that comes up, we have different isotypes, but the gene for all of it is arranged on the same chromosome. So, this is some just a factual detail that you might want to remember. Okay, now finally coming to how they are arranged, right? So let's just see here. So this is a diagram from QB, and this talks about the arrangement that you see in mouse model, right? Let's just discuss how we see it in humans, and then we will just try to see this diagram and see if we are able to correlate things, right? So if you see a double-stranded DNA for the kappa light chain we are talking right now, we have a variable region here, again, here. So there are multiple V regions that are arranged in succession on a linear strand of DNA, right? With each variable region, we have its respective leader sequence that we just said. We just said that we have a short stretch of a nucleotide called as the signal sequence or as the leader sequence present upstream of the variable region. Similarly, we also have, so how many for a kappa light chain, how many such V regions can be there? It's in humans, it's somewhere around 40. So you can have V1, V2, V3, V4, V5 and so on for until V40, that they can be arranged in this linear fashion, each having its leader sequence, right? So you, all of you understand what is intron and what is exon. If there's anyone who doesn't understand what is an intron and what is an exon, you can please either raise your hand or unmute yourself. Okay, good. So this leader sequence, the V sequence, all of this is exon. Exon is what comes in the protein that is ultimately expressed from the gene, right? We have certain intervening sequences, which is also called as the introns, and those are non-coding sequences, and we will see that they get spliced off. So as we were saying that they have a chance of recombining, recombination is going to ensure only one V to get like randomly selected, and it gets a chance to recombine with either of the J segments, right? So what is J segment? Again, a short stretch of DNA that we are we are naming it as J segments. And how many such J segments are possible for a kappa chain? We can have such 5 such J segments separated by certain intervening sequences, right? So if you just see, and then again we have a long intron which is separating it from the constant region of kappa chain, right? So once constant region corresponding to kappa chain is present, but if we just look at the lambda chain, we can have multiple such C regions, and only one of them can get a chance to recombine, right? The difference between these two, so most likely, So what happens while the process of rearrangement occurs? The first the first chance is given to kappa chain for kappa to undergo rearrangement. This and if it forms a productive rearrangement, which means a rearrangement that can actually bind antigen that gets selected, we have the antibody molecule wherein the light chain is a kappa light chain. If for some reason, by all sorts of possibility, this B cell fails to develop a kappa chain with a productive rearrangement, it is then that your lambda chain gets a chance to rearrange, right? So most of the cells that we see in circulation, most of the centrocytes, they are going to have kappa chain. Lambda chain is only going to be present in a very few cells. The reason behind it, I just told you that first chance is always given to kappa chain. If after multiple rearrangement by certain mutations also, the affinity is not sufficient, the rearrangement is not coming productive, that cell is triggered for apoptosis and further division is going to select for cells with lambda chain. The only difference that you see between these two is that we again have multiple variable regions arranged in a linear fashion, and we had somewhere around 30 such variable regions is possible, a long intervening sequence. And then we have J chains, J lambda chain, which can be present with C1 or C2, C3. So a J lambda is possibly there with all the different C regions that we see in case of lambda chain. So just how the organization of kappa and lambda chain is there, right? Just some details there. Apart from that, did I not keep the heavy chain rearrangement in humans? Okay, we'll just try and understand from here because it's somewhat still similar. So it's the same thing here for mouse, but so the number of base pairs that can be separating it, the n number here. So those are some things that can vary between the mouse and human chain. Or where did I keep the heavy chain arrangement? Okay. No problem. So this is how your heavy chain is arranged. Where again we have the V chains arranged linearly. How many of them? So we do not have a number that is possibly there even for humans. Then we have what we called as D regions, right? So I am just mentioning for human. So for mouse, it is somewhere D1 to D13. For humans, this N number can be up to 20. So we can have 20 such D regions present in succession. Then we have your J regions present. For mouse, it's J1 to 4. For humans, it is 6. Right? These are the numbers that correspond to humans. So V, D, and J. So this D region was not present in the light chain arrangement. We have D region arranged in your heavy chain. So we have V, D, J, and then the closest possibility is we have so over here the C mu that corresponds to IgM and C del that corresponds to IgD. They are present in close succession with each other. So like very close by each other when it is arranged in a linear fashion. The number of base pair between this is comparatively much lesser than the number of base pairs that you see between the other possible uh, constant region which is arranged on this heavy chain DNA, right? So I hope this is clear that two things that has or three things that you must understand from this overall diagram is that lambda chain and kappa chain is being encoded by genes present entirely on different chromosomes. For both the light chain, we have V, J, and C regions. But for heavy chain, in addition to V, J, and C region, we also have a D region between V and J. Third thing, is that all the different isotypes determined by the constant region of your heavy chain, all such constant regions are arranged on the same chromosome. And it is only by that which recombination brings together which all constant region that determines that which antibody is being produced by the cell. Now, uh, so let me just clear here that so when we have been talking about class switching, initially we see that it's the IgM and IgD that is produced. In class switching, your VDJ chains, they remain the same. It's only the constant region that that's again gets a chance to recombine, and here we can have them coming together with either of these constant regions, right? And which constant region is participating, that determines the type of antibody. We'll discuss this further. But in class switching, the variable regions do not change. It is just the constant regions that change because of alternate splicing and recombination that happens, right? Okay. So how this we just saw how the immunoglobulin genes are organized. Now let's come to how the process of rearrangement is happening. So rearrangement of B genes produce antigenically committed B cells. How? The variable region is the point which had, if you remember, the variable region has CDRs, which are the hypervariable region, and the antigen binding is actually facilitated by the CDRs present in your variable region. So, whatever is the amino acids present in this variable region, that determines which antigen can be bound by a particular B cell. So the rearrangement fixes the variable region of your B cell, and that is how makes a B cell antigenically committed. So, and in which cell this rearrangement happens? So if you see here, uh, we have the yeah, so right here we had a lymphoid cell which has the D and J chains of heavy chain coming together into a pro B cell. In this pro B cell, the VDJ recombination of a heavy chain is happening, and that is when a pre B cell forms. Now, once the heavy chains are assembled, so during the process of heavy chain assembly, we have something called as the surrogate light chain that gets a chance to arrange on this pre-B cell temporarily till the time the light chain rearrangement happens. So first we have the heavy chain rearrangement happening. DJ rearrangement is making a pro B cell. VDJ rearrangement is making a pre B cell. So when the V chain joins the D and G, we have a pre B cell. Then we have the light chain arrangement, which makes a immature B cell. Okay. So coming back here, so this rearrangement of the variable region genes, if you see there, we did not say about the constant regions rearrangement, because in light chain it do not make a meaningful sense, because because it's either kappa or lambda, and that selection has already happened before the variable of kappa and or the variable of lambda has joined together. But for your heavy chain also, which constant region has come together, that essentially do not have anything to do with the antigenic commitment of your B cell. So your mature, most importantly, what is actually happening in the process of production of mature B cells is the rearrangement of V genes, the variable region genes. And once we have this rearrangement, once this has happened, it produces an immunocompetent B cell which can bind an antigen. This process of rearrangement is an entirely random event. So the antigenic specificity is determined randomly. So the body just tries to bring different combinations together to bring about the diversity that we have been talking about, right? This is how the process of rearrangement is actually occurring. So we saw the multi-gene arrangement that was happening. We discussed two things. That one is multi-gene organization and the process of IG genes rearranging. This is how the process of rearranging happens. Okay. First, we'll discuss about what is happening for your light chain, and then we will talk about the heavy chain. So in the germ line, we have discussed about kappa. So they have more often you will find the kappa chain discussion whenever we are talking about the light chain gene rearrangement. So in the germline, this kappa chain DNA with different V regions possible, they come together. How do they come together? So this is a 5 prime and 3 prime. There is a 3 prime region that can combine to 5 prime of either of these J chains, right? So let's just say this first one has now the come together with the third J gene segment. So first to happen is the VJ joining. This VJ joining that happened here, what is it consisting of? It is consisting of a leader exon. So leader exon, again, it's not the part of the protein, right? The leader sequence only helps to spoon in the this particular transcript, the final protein that forms. It helps to like just spool in that. If you have some background from cell biology, you would know that entering of a protein into the endoplasmic reticulum, it requires a signal sequence. This signal sequence, once its job is done, once the protein is inside the endoplasmic reticulum, the signal sequence is chopped off, right? So this leader sequence is present. So in the primary RNA transcript, we have this leader sequence. So all the chains that do not get a chance to join, so they are now cleaved off. We do not see them anymore. We have them spliced off, and this productive combination is maintained into a primary RNA transcript. So from 5 prime to the 3 prime direction, if you see, we have a leader exon, we have a non-coding intron sequence, we then have the joined VJ sequence, and now we will have now this has to join to a C segment, right? So for kappa, we have just one C. So this joining happens. There is polyadenylation and RNA splicing, essentially the terms from molecular biology subject. But so the way this polyadenylation happens and the way the RNA splicing happens, that determines the process of this joining. We ultimately have this A tail because of this polyadenylation. It's a sequence for that helps in initiation of translation. And ultimately, from this mRNA, we have a nascent polypeptide, which is now ready. It enters the endoplasmic reticulum where this leader sequence is removed, and the productive chain that forms has the V, J, and C region. Has the V, J, and C region now. Now, this light chain, this is a ready to join light chain, which what it is going to join? It is going to join with the heavy chain inside of the endoplasmic reticulum only. The heavy chain that has separately undergone this process of rearrangement and recombination, and together they will form the immunoglobulin molecule. So what you see here, what exactly happened in this joining, it's you can visualize it as a cut and paste, random cut and paste of the different regions with other different regions is going to give rise. We have different permutation combinations happening, because of which the diversity is happening. Now this process of cut and paste is actually in scientifically it is called as VDJ recombination given so this entire thing was this person S. Tonigawa and N. Hozumi. Tonigawa was also given the Nobel Prize for this concept. He actually worked to show that it can rearrange. He essentially did it with I think it was Southern blots that he used to show how this is happening, what all different combinations are coming up and everything. But today it's comparatively easier to do with all the sequencing techniques that we have available. But he laid the foundation for the discovery of VDJ recombination that happens during the process of B cell maturation inside the bone marrow. There's an enzyme complex that mediates this recombination, because any sort of cut and paste that you see happening inside of a cell, we have enzymes that catalyze every such cellular reaction. Which enzyme? So VDJ recombinase, which essentially has two lymphocyte specific proteins. So if someone says that a protein that is specific to lymphocyte, we have RAG1 and RAG2, recombination activating gene, and they are going to get translated into RAG1 protein and RAG2 protein. These two proteins, along with other proteins involved in DNA DNA repair enzymes. These proteins, along with DNA repair proteins, are enzymes. So these are also enzymes, DNA repair enzymes. All these enzymes together bring about this process of VDJ recombination, right? We'll see the mechanism underlying that as well. But let's just complete the same discussion for heavy chain gene rearrangement. So if you understand the light chain gene rearrangement, it's everything is the same. Over here, we have the germline HDNA, which possibly has all the different introns put together, arranged together. First to happen in a heavy chain is the DJ joining. So this DJ joining is going to bring about one such joint. Then we will have a V joining to this DJ joining. We will again get a VDJ joining happening here. And if you look here, we will similarly have from the 5' to 3' end, we have a leader sequence, which will again not be present in the heavy chain peptide, in the heavy chain protein that forms. Some introns there, a VDJ segment together, again a stretch of introns. Along now, the joining of C segment will happen. So since so these two sides we just discussed is the closest to this VDJ junction, they get a chance to get transcribed together. So what forms essentially first is a transcript with both C mu and C del. So we have the primary RNA transcript. So rearrangement happened here, which in a developing, in a maturing B cell is going to completely rule out these constant region. The first to occur in the primary RNA transcript is C mu and C del, right? When a B cell is undergoing the development from a pro B cell to a naive B cell, this is what is happening. For whenever only in the presence of antigen, you might have, so we'll come to that part later, but you might have these regions joining to some other constant regions, and that is going to give you the particular isotype that we have been talking about, right? The primary RNA transcript has both C mu and C del. Now this, similar to what we saw for light chain, it has to undergo polyadenylation and RNA splicing. This is going to give two mRNAs within the same cell, which get a chance to translate into this nascent polypeptide having C mu as well as C del. And ultimately, this leader sequence gets cut off, and we have two heavy chain being produced inside the endoplasmic reticulum of the same cell, one of C mu type and the other of C del type. Both these chains can recombine with the kappa chain, which is most likely to be present. But if we have a lambda chain, the lambda chain gets a chance to come and recombine with either of these chains. And that is how a fully functional antibody molecule is assembled. So far, is there anything that is not clear? I'll I'll give you all 5 minutes to just stay with this. And if there's anything that is not clear before we move forward, we'll first try and discuss that. Okay, all clear. How come?
Excuse me,
Mhm.
ma'am.
Yes, yes. I was just about to ask, how come Shamika is so quiet today? Am I going wrong somewhere?
No, ma'am, I'm having some doubts. So,
Please go
regarding
ahead.
the whole concept that you have explained right now, so will it be possible for you to
Yeah, yeah, please go
will
ahead.
be able to answer me all those, or will you will be doing that in end of the session?
I think you can go ahead because when we discuss some doubts, we essentially have to discuss the mechanism of all of this, and that will not take up much time. And also doing some doubts in between can help the others to like wake up a little. So you can just go ahead with it.
Okay, ma'am. So, ma'am, the genes like we have shown is gene, V gene, and D and J gene. So these are all present on all the. These are all present in the heavy chain and the light chain.
Yes,
So, ma'am, you have told that the D gene is present on heavy chain only and not on the light chain. So, is
yes.
there any other gene which, like from C, V, and J gene, which is present only on any one, or all these
No.
three are present on all of these?
See, so when we say about V chain or C chain, So the V gene for either the light chain or the heavy chain, they are encoding the, so it's going to code for the N terminal of the antibody molecule. N terminal, if you remember, is where the antigen binding site is there. So we have different variable regions which can come together with either of the J segments, which together can again come together with either C kappa if you're talking about light chain, or with any of the C segments present for the heavy chain. And that is how the heavy and light chain molecule is assembled. Only this D segment is in addition present in heavy chain between the V and the J region. The variability that comes in the antibody molecule is because of this V gene segment, because that is the segment that encodes for the N terminal of the protein where the antigen binding site is present. Did
Okay, ma'am.
I answer
Yes,
your?
ma'am.
Mhm.
And ma'am, these are the gene sequence which are present on the present inside the DNA of the B cell. Am I right in this?
Yes, so here we have this germline. So in the immature B cell, or say the hematopoietic stem cell, once it is selected for the B cell lineage, it now so if you remember, if you would know that we all cells of our body have all the genes present, right? So this
Yes, ma'am.
H chain and the J and the kappa chain or the lambda chain, it is switched on once the commitment from hematopoietic stem cells to the cells of the B lineage happens, right? So as we saw here, this lymphoid chain, as soon as it undergoes this D and J joining of heavy chain, that commits it to becoming a pro B cell. And all the subsequent joining is happening so that this pro B cell can convert into a naive B cell. So the process of rearrangement is going on as the cell is undergoing cell division and differentiation.
Ma'am, this rearrangement is different from the recombination that we see in the meiosis, right?
Yes. So that is a question that
Yes,
initially
ma'am.
came up. I hope this class answers that query.
Yes, ma'am. And ma'am, this L region, leader axon, is present only in front of the variable region so as to suppress or repress the activity of the specific variable region gene, right now?
See, the function of, so if you would see the RNA transcripts that are forming for heavy chain as well as for light chain, the L sequence is present at the 5' terminal. So this is a concept from cell biology where that I was saying that for a polypeptide, because immunoglobulins, so this will release a polypeptide which is somewhat in a straight configuration, right? But for the immunoglobulin gene to get assembled, it has to undergo folding. This folding occurs in endoplasmic reticulum. The only function of this L sequence is to target this protein inside of the endoplasmic reticulum. उतना ही काम है लीडर सीक्वेंस का बस. कोई additional, so it is not there in the ultimate polypeptide that is in the ultimate protein that is formed in the ultimate light and heavy chain. It is not present because its only function is to come inside. This is not it has not got anything to do with the signal sequence, the recombination signal sequence that we will just discuss, which guides the process of recombination. The leader sequence is only there upstream of the variable region, because when a V is going to combine with D, J or whatever, ultimately the 5' end is going to have this leader sequence. And this leader sequence is required for it to enter the endoplasmic reticulum. It is not going to guide the recombination.
Okay, ma'am. And ma'am, the foldings to the polypeptide chain is provided by the endoplasmic reticulum.
The folding of any polypeptide is always happening inside the endoplasmic reticulum. So including the immunoglobulins.
Okay, ma'am. So, ma'am, after the nascent polypeptide is get matured, the L region or the L gene is get removed from the
Yes.
heavy chain or the light chain, right? Because its function got
Yes.
over.
Yes. Yes.
So, ma'am, the antibody molecule is having VDJ and constant region only, which is making up the whole antibody structure inside the
Exactly.
transmembrane, right?
Yes. 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 So if the B cell has to secrete out the antibodies, if it's a plasma cell, if it has to produce antibodies, the transmembrane domain is going to be cleaved off. That's it. Apart from that, which antibody is being produced, that is going to be determined by this VDJ recombination.
Here the you're talking about the isotype, right? IgA, IgD, and so
Mhm.
on and so forth. So they are determined by these rearrangement, right? So here, if
Yes,
we see that C delta is present, so it will be forming IgD type of antibodies. Am I right?
yes, yes, yes. So whichever C region uh, gets recombined, if it's C mu, we have IgM. If it's C del, it's IgD, and so on.
Okay, ma'am. And ma'am, you have told that right now, that plasma cell, right? If more and more antigen is encountered in our body, and plasma cells need to be synthesized in more amount, so the plasma cells will going through all these processes again and again, or it will
No.
be like just a manipulation of those a preformed once a pre plasma cell is formed.
See what happens. So, just take a minute to read this. The rearrangement of heavy chain constant region generates this IgG class, but without any difference in its antigenic specificity. Why so? Because in a plasma cell, this combination is fixed. Which, so if we have say 1 to 20 variable regions possible, which of the variable region has got a chance to come together in this mu chain? This is fixed. So if we say, maybe if this one got selected and say, so we were discussing it like this, my bad, I forgot. So this is J3 maybe, and maybe D7 got a chance to come together, right? So it is going to be V1, it is going to be V1, D7, and J3. Now, if a class switch has to happen, or if the membrane bound antibody has to transition into a secretory form, so if the B cell has to now become a plasma cell, it is going to only change the make of C region. Maybe it can become C gamma 1, right? But this combination remains fixed. So, in the process of class switching, or while more and more plasma cells are differentiating, because this is the part that actually determines the process of clonal selection. So, the cell is undergoing differentiation, but if you remember, in the antigen dependent phase, so in the antigen dependent phase, your antigen is driving the entire process, right? So, this antigen has selected for this particular combination of V, D, and G. The only thing that is now we want to decide based on which effector function we want to bring about. We can only change our C region for your heavy chain. But clonal selection is going to only happen for the particular selected combination of VDJ for heavy chain or VJ in your light chain.
Ma'am, actually, there are some, some related terms which are related with each other. So I'm getting confused in those terms, right, that you have used, right? The first one is the clonal selection. Then previous classes you have used the antigen specificity, and in today's class as well you have somewhere wrote that in these diagrams as the labeling. I'm not remembering that exact word, so I'm getting a bit confused in those words. I know that
Okay.
there is a slight difference between them, but actually I'm not getting cleared with these terms. So will you be able to do that?
Okay. We can see this diagram. So this is your the baseline, right? This is the framework which has almost everything there. Let's just try and add more things to it, right? And you try to just follow it with me. And it would be good if you like also have a chance to write it with me, because that helps in clarity. Okay. So if you just see here, in the so we have a hematopoietic stem cell here, which can differentiate into any lineage. Okay. Clonal selection comes much later, right? We first have a simple cell which is going to divide. If it has to become a B cell, if it ultimately has to become a B cell, this hematopoietic stem cell is going to express certain markers on its surface along with the Ig alpha and beta molecule, right? Whichever cell so this cell is also going to have all the different genes that are also going to be present in every cell of your body. If the cell if the gene that is now getting expressed are these particular genes, this cell is known as a pro B cell. In this pro B cell, the VDJ selection, the recombination, and everything is happening, right? So whenever we have the heavy chain, the first to happen is your heavy chain rearrangement. And since the naive B cells always have IgD and IgM molecules coming up, what do you think will be the ultimate result of this heavy chain gene rearrangement? So let's just say it can have V1. So we were talking about V1, D7, and J3. So of all the different combination that can be produced, let's say that we have an antigen that can only bind if the make of the variable region of a heavy chain is like so. Okay. So it's not like we will only have one free B cell with this particular combination. You're going to have many B cells randomly forming. It is a very random process, and whichever B cell is produced with this combination by a matter of chance, we have this antigen which can only bind some heavy chain which is of this make, right? If at any point you feel lost, you can stop me. Take care. Now, once this is formed, this is for your variable region. But your constant region, since we know it can only be IgM or IgD, it, it can either be C mu or it can be C del. Okay. C mu and C del, we saw that initially the primary transcript that forms, it has C mu and C del in close association with this combination of VDJ. There happens splicing and polyadenylation, which ultimately results in in the same cell having this variable region with C mu make as well as this variable region with C del make. Both these cells, both these heavy chains are now going to be expressed on the pre-B cell, right?
Excuse me, ma'am.
Mm-hmm.
Ma'am, here you have told that any of the combination can be possible, like V1, D7, J3 can combine with C del as well, or it can go with the C nu as well, or it may have both.
It will, so any recombination that can happen. So it can be, what I am referring to is, it can also be V2, DA, sorry, It can also be
That I
V2.
understand that
Yeah,
the prior portion can be anything, but you are telling here that C new and C del. So I'm thinking that it's
this cell
having
is
both.
going to produce both. Initially, that is what I am saying, that we just saw that in with this VDJ segment, we have the C mu as well as the C del. These are very close. It's only 4.5 kilo base pairs apart compared to, so over here, see, over here.
Mom, you mean that they both are traveling together?
In a way, yes. So over here, if you see, your C mu and C del is very close together, right? So after the recombination that happens, we have a primary transcript that has both of them together. There happens a process of polyadenylation and RNA splicing where we have the same B cell producing this as well as this. So consider that, so at from this point, this can combine with either this or this. But it's not like I one cell is only going to produce either this or this. You have multiple such RNA transcripts, because it's not like you will have, so this is a rearrangement that happened and this transcript is going to be transcribed multiple times. So this DNA is going to be acted upon by your RNA polymerase multiple times and multiple such transcripts is going to be produced. Might be that one transcript, we have this point joined here, and that's how this joining happens where the VDJ is with C mu. Whereas in another possibility, some other transcript can undergo a differential splicing. The splice that has formed is different. And now we have the VDJ joined with the C del segment. So ultimately, in the same cell, you have both the heavy chains present. Is this now clear?
यस मैम। तो मैम यहाँ पर जैसे सी न्यू एंड सी डेल वो दोनों
Mm-hmm.
साथ में ट्रैवल या फिर साथ में तो नहीं रहेंगे ना? एक सेप दो ऑप्शन है कि दोनों में से कोई भी एक फॉर्म हो सकता है, एंड ये आरएनए स्प्लाइसिंग जो है दिस प्रोसेस विल डिसाइडेड, राइट?
Yes. So in the process in the pro B cell state, okay, in the pro B cell, before your V, D, and J joins to a C segment. So the decision of joining is in for a heavy chain is happening at the stage of pro B cell, right? When this pro B cell becomes a pre B cell, you will have some transcripts of VDJ joined with C mu, and that's how you will have this pre B cell expressing this. And you will also have some transcript joined with C del, and you will have the same B cell expressing C mu as well as C del.
सो मैम एक इम्यूनोग्लोबिन आईजी मॉलिक्यूल में सी न्यू एंड सी डेल दोनों मिलकर के एक आईजी मॉलिक्यूल बनाएंगे कि सिर्फ
नहीं,
एक ही?
वन, आइदर ऑफ देम। तो हम लोगों ने ये देखा था कि जब एक नाइव बी सेल है, we have two antibody molecules on it, right? One is one is of IgD type and the other is of IgM type. So this is your pre B cell. So
सो
this
मैम
cell
यहाँ
has both.
पर सो मैम यहाँ पर इसमें से सी डेल्टा वाला पोर्शन जो है वो एक इम्यूनोग्लोबुलिन में पार्ट हैवी चेन
Yes,
फॉर्म करेगा,
yes,
एंड सी यू वाला जो है वो आईजीएम में लेट सपोज अगर हम अज्यूम करें तो उसमें हैवी चेन जो है वो फॉर्म करेगा। सो
yes,
दैट्स वाई यहाँ पर दो पॉसिबल कॉम्बिनेशंस जो हैं वो हम उससे कम अप हो रहे हैं। सो एवरी
yes,
टाइम
yes.
यही सेम प्रोसीजर फॉलो होगा?
If in the state until the state of a pre B cell, yes. But when a plasma cell has to be formed, when the antigenic clone, अब हम आएंगे क्लोनल सिलेक्शन पे, जब क्लोनल सिलेक्शन हो चुका है तब class switching comes into picture. In this process of class switching, now instead of ये प्राइमरी ट्रांसक्रिप्ट बनने की जगह we will have certain other combinations possible. So यहाँ जो पहले यहाँ from pro to pre B cell का जब state है, जब antigen encounter ही नहीं हुआ है, उस point पे joining हमेशा यहीं पे जाके होगी ताकि आपका ये ट्रांसक्रिप्ट बने. बट जब एंटीजेनिक सिलेक्शन हो गया है, जब क्लोनल सिलेक्शन होना है, उस पॉइंट पे दिस ज्वाइनिंग कैन विल नॉट प्रोड्यूस आईजीएम एंड आईजीडी. इट मतलब इट कैन प्रोड्यूस आईजीएम एंड आईजीडी, but primarily the immunoglobulin that is getting produced in our body, we saw in the last class, is of IgG type. How is that happening? It's not like your okay, no problem. In that case, it is not like we will have this transcript first and then something else is going to come and join. No. Instead of this transcripts formation, we will have this segment joining somewhere over here. When it joins over here, this entire region is spliced off in a loop, and we will have the transcript that has L, V, D, J, and say C gamma 1. That's how the process of class switching happens when we are talking in terms of antigenic selection. Any better? Okay, let's come back here. ठीक है? We digressed off. ठीक है? हम one by one चलते हैं. Over here, हमने देख लिया कि हमारे पास of all the different combinations that can form, we have antigen present over here which can only bind to this make, and that's why all the cells we are now right now discussing about this particular combination of VDJ. So जब ये सेल बन रहा है, इसको नहीं पता है कि इसको जाके यहाँ एक एंटीजन मिलेगा जो इससे बाइंड कर सकता है. ठीक है, वो बॉडी तो रैंडमली सेल्स बना रही है, बट हम लोग अभी ये देख रहे हैं कि इस पर्टिकुलर कॉम्बिनेशन के साथ सी म्यू और सी डेल बना. सिमिलरली हमारे पास लाइट चेन रीअरेंजमेंट भी आएगा इन रोल, and maybe that light chain again, this rearrangement can also be of different types, right? Multiple V and J segments can come together, but let's just assume the example of whatever, let's just say V2 and J9 has come together. So this antigen is of variable regions of heavy, so there is no D in heavy chain. जिस भी एंटीबॉडी का वेरिएबल रीजन लाइट चेन का ऐसा होगा और हैवी चेन का ऐसा होगा, ये एंटीजन उसी से बाइंड कर सकता है. It's a matter of chance that we have a heavy chain which has this specificity, and it has also got a chance कि उसका लाइट चेन जो रीअरेंजमेंट हुआ है उसमें भी यही कॉम्बिनेशन आया है. ठीक है, तो इस पॉइंट पे क्योंकि लाइट चेन रीअरेंजमेंट हो चुका है और उसका वेरिएबल रीजन डिफाइन हो चुका है, ऑल्सो इससे पिछले स्टेप पे ही हमारे पास हैवी चेन का भी रीअरेंजमेंट हो चुका है, तो उससे भी एंटीजेनिक डिटर्मिनेशन मेरा हो चुका है. अब यहाँ जो इमेच्योर बी सेल है, इट इज कॉल्ड एंटीजेनिकली कमिटेड. ठीक है, यू सो देयर आर थ्री टर्म्स यू वर टॉकिंग अबाउट: clonal selection, antigenic commitment, and VDJ recombination, right? If I remember correctly. So antigenic commitment occurs as a consequence of heavy and light chain rearrangement. Okay? Because once you have the rearrangements done, that has already defined that which antigen it can bind. That definition has already happened because whatever recombination had to take place, it has already taken place. These two molecules have come together, and this point, this immature B cell is called antigenically committed. Is it clear so far?
Yes, ma'am.
Okay. Now antigenic commit. So it's not like so along with this immature B cell, which this combination, you will have multiple other immature B cell which can have some other combination of variable region, right? But it is the presence of this antigen that triggers for clonal selection of a cell only with this particular make. So we will let's just say that we have, so I hope you understand that it's the same combination here, And it's again the same combination of variable region here, right? Now, this particular cell, along with other cells with some different combination, all of it comes into your peripheral lymphoid organ. But the antigen waiting here, it is of a shape and kind where only the antibody molecule combination of this type can bind to this antigen. So now I have from among all the different B cell with different types of antibody, this particular B cell, see, recognize it is now, so your follicular dendritic cell there is presenting this antigen on its surfaces. All the different cells are binding, trying to bind to this antigen. But only the cell with this combination of variable region is going to get a chance to interact with it because it is committed to this particular antigen. इसने जिसको commit किया उसको वो मिल गया, तो वो उसको पकड़ के रखेगा. अब जैसे ही उसने उसको पकड़ के रखा, उसने इस एंटीजन को प्रोसेस किया और इसमें क्या आता है, एक्टिवेशन हो गया बी सेल का. जब बी सेल एक्टिवेट होता है तब उसमें क्लोनल सिलेक्शन होता है. क्लोनल सिलेक्शन कैसे होता है? ऐसा नहीं है कि आपका यही सेल, this cell is not going to, so your mature B cell, it is now a centrocyte. Here it was a centroblast, right? But here it is a centrocyte. This cell is not going to say again divide into daughter cells, because if this is dividing के लिए भी आपको क्या चाहिए? You initially need all the cell cycle re-entry for division and everything. The only possible thing what happens is that when this cell, okay, just give me 1 minute. Let's just ensure that what I'm saying is correct. तो यहाँ बना हमारा सेंट्रोसाइट. ठीक है, दिस सेंट्रोसाइट इज नॉट डिवाइडिंग, राइट? बट एट द सेम टाइम बॉडी ने सिर्फ एक ही तो ऐसा बी सेल नहीं बनाया है जिसके पास ये कॉम्बिनेशन है. मतलब here we are talking in terms of millions, right? Number of cells with different combination, and there might be 100 such cells already which already have this combination. When this combination of antibody gets selected, this this selection is going to lead to clonal selection or clonal proliferation, wherein all the other remaining centroblasts are now going to differentiate into this particular cell type only. So the number of the million of cell, अगर मेरे पास पहले ये cell से 100 present थे, within no time I will see that I have thousands of cells of this particular same type. ऐसा भी हो सकता है ना कि आपके पास जो lymph node है उसमें 10 सेल्स थे इस टाइप के, ठीक है, कुछ बाकी के 10 सेल्स बॉडी में कहीं और सर्वेलेंस में प्रेजेंट थे. So once your antigenic selection is happening, activation is happening. Again, remember in the process of activation, we have a role of T helper cells. When the T helper cells are coming in, it is going to produce cytokines. These cytokines from other places in your peripheral tissue is going to attract the cells of this particular combination to reach the site of antigenic challenge. And all together, so you have certain cells migrating in there. You have multiple centroblasts differentiating further to produce this combination. So we have this बहुत ज्यादा division trigger हो जाता है आपका, इस combination का multiple selection हो जाता है, and that's how we have so many cells of this particular cell receptor make coming into place, and that's how the process of clonal. So this overall process is called clonal selection, where we have multiple, instead of the initial 10 which encountered the antigen, you have thousands and millions of cells of this particular type now taking care of the antigen together. So this process is called clonal selection. The phenomena is called clonal selection. which only happens in presence of an antigen. Now, if you would have had some other antigen, which could be bound by some other antibody, uh, other combination of variable region of the light and heavy chain, so therein you would not have seen this cell number multiply. So that's how antigenic selection is the part wherein only the cell with a particular make of antibody gets a chance to proliferate and participate. इज दिस क्लियर नाउ? ठीक है, अब हम आएंगे क्लास स्विचिंग पे। अब बहुत सारे सेल्स ऑफ द सेम टाइप बन तो गए, बट हम लोग पढ़ेंगे इस बारे में। जो सी रीजन है हेवी चेन का, दैट डिटरमाइंस कि which function of antibody is going to be taking care of the antigen. If you remember from the second class, I think something called as neutralization is possible, something called as complement activation is possible, something called as opsonization is possible. So these different biological functions that are brought about by the antibody is essentially by binding of the C region of the antibody to the different receptor present on some cells from the innate immunity, right? So again, that is to understand better about it. I think we have to wait for another 2 classes, and then we will understand about what all effector functions are being performed by different antibodies. For now, just remember that how is all these types of antibodies different from each other? The only difference lies in their constant region. The constant region actually determines which effector function, either opsonization or complement activation, which function comes into play is determined by the constant region. So body would want that you have a multi-pronged approach taking care of the same antigen. So it might want complement activation, it might also want neutralization, it wants optimization, opsonization, and how is it going to bring about it? It needs to switch the isotype. Again, so how the switching of isotype is happening? That the variable regions make is going to be cell. The body knows the selection is only going to occur in favor of this particular type of variable region. But the heavy chain produced here, the C chain that is produced here can be of a different type now. So it can be of C gamma, C alpha, it could be of C epsilon based on whatever the body's instructions is. It could be of all the different types. So it is actually known that in presence, so the first to come in picture is IgM, of course, because it is already stationed on decorated on a B cell, and whenever a plasma cell is formed, the first instinct of that cell, because it already has IgM, so those are already stationed military. The first to come in picture is IgM, but the most effective function in clearing out antigens is of IgG. So that is what we discussed here. That whenever we have a primary response, we have IgM, right? In the secondary response, we discussed that a heightened immune function is observed and this lag period is also delayed. But people have seen that the response, the amplitude of IgM molecules that is produced is roughly the same. The IgG molecules take over. Why so? Because the effector functions that IgG can mediate is the most effective in clearing out the antigenic challenge. So there is the role of class switching. So how is this happening? Because now when the differentiation and development is happening, instead of the recombination of your VDJ genes with del and mu, we have the recombination happening with either of the gamma genes, because again we saw that in Ig we have Ig1, Ig2, Ig3 and so on. So we based on small amino acid differences in the constant region of these, we have certain subtypes, right? So based on whichever joining is happening, we can have the variable region being constant. It's only the C region that is different now. So that's what I'm saying. You have to bring everything together to understand what exactly is happening. So it is a lot of terms, and it is a lot of thing that is happening together. We are trying to understand everything one by one, but it is important for us to understand that all of this is actually happening simultaneously. Now tell me what is not clear.
So, ma'am, class switching is happening after the encounter of the antigen and when
Yes,
the cell is wanted to divide and want to clear that antigen. So, in
yes,
that case, class switching is taking place, right?
yes, yes. So class switching only occurs in presence of antigen. Okay?
Okay, ma'am. And ma'am, so we can say that from the very beginning, that is from the development of the hematopoietic stem cell into the B cell, so the whole process is repeating again and again as to clear the
Yes,
antigen, right? Because
yes, yes.
so ma'am, different IgG types will be forming, right? To clear
Different
that
IgG
antigen.
type would form if the requirement is such that we want the Ig, we want some antibodies in the secretions of the body, IgA is going to form. So whatever is the requirement of the body, class switch is going to happen in that direction. What determines this class switch? So I'm saying that whatever is a requirement of body, but there is something that is going to guide this. It's going to tell the cell that this is the requirement of the body. Who does it? The cytokines that are being produced from the T helper cells that is interacting with the B cells throughout, right? So
Okay, ma'am.
we will
Uh,
not
so
get much. Bolo bolo.
Ma'am, in the class switching case, the variable region, right? So the
Mhm.
variable region portion remains the same in all those again and again forming B cells, right? We can say
Yes,
so.
it is like so. The variable region remains the same. The combination of VDJ is the same in class switching. It's only your C region that is getting a chance to differentially recombine.
So, ma'am, in that case, we can say that there is no rearrangement is taking place in variable region.
Yes. So it's like so when the cell is dividing, your division is maintaining. So this VDJ has now come together, right? So whatever is the possibility of introns and exon there, how is this recombination happening? There is some introns and exons that are getting spliced off. We have the RAG1, RAG2, which is mediating all these cut and joinings, right? Once they have come together, there is no signal that can guide for the recombination of it, because whatever is the introns present between it, they have already been chopped off. Only because the last to join is the C region, right? So VDJ joining has already happened. Now this is fixed. This can now join with either of the C regions. It's the last to join, right? So there it has this right over here. Since this is fixed the first. What happened to this? So over here, this is, I'm going to write on it. Okay, this is fixed if you see here, right? Now the last, so VDJ combination happens first and then different C regions can come and join. So, so after antigenic commitment, we have this combination fixed. In class switching, only this can now have introns and all by using which it can join to any of the constant regions.
Okay, ma'am. And ma'am, you have shown that 12, 14. So these are the chromosome number or the number of genes which are present on that chromosome.
Chromosome number. This is chromosome numbers. Okay.
Okay, ma'am.
Okay. So anything from anyone else that you might be thinking now after all this discussion? It's very important to have someone to ask questions, because I think it also clarifies a lot of things, and it was a very valid doubt that so many things are happening. I'm all confused. That is the case all the time. So that the bottom line is we are understanding everything one by one, but everything is happening simultaneously, right? Okay, just give me 1 second. I'll plug in. Okay, now that we understand all the recombination and everything that is happening, we'll try and understand that what exactly is the mechanism mediating it. Now, if you look into QB, it gives you a diagram that looks somewhere something like this. But so I think this is looking a little more complicated, owing to the fact that it is trying to show you what all folding and crossing over of the DNA and everything is happening. So we'll try and understand what is happening using a simplified, more simplified diagram, and then we will try and see what is happening in the diagram in the reference book. Okay. So before we get into the mechanism, there are certain facts that we need to understand. The first thing is, in front of a V, D, and J exon, upstream of it, we have upstream as well as, so I think it's upstream of it, we have, so it can be, the case can also be downstream, right? So over here, if you see that this is a D sequence and there is something upstream of it also, there is something downstream of it also. So flanking is the right word, that either side possibly we can have what we call as recombination signal sequence, RSS in short. So RSS, what it has, this is a signal sequence, which means it is going to signal something. What is it signaling? It is going to signal the binding of the RAC complex that we just mentioned. How is so whenever we have a signal sequence, there is certain something called as consensus. Consensus means consensus, conserved. These words mean that in all the different cells, in all across the different phylum, you will have a conserved sequence. It's the same sequence of DNA base pairs, the nucleotide base pairs, arranged on either sides of your V, D, and J exons, right? These signal sequences can either be of nonamer or heptamer, which means it can either be 8 base pair or it can be 9 base pairs. So, and they are separated from each other by either 23 base pairs, which could be any base pairs. So this is conserved. So the sequence of CACAGTG, or this particular sequence, this particular arrangement of different nucleotide base pairs, this is conserved. There will always be CACA, GTG. It cannot be CT, CN, and that is not going to be called as a conserved sequence. So we have conserved heptamer and nonamer sequence separated from each other by either 23 base pairs or 12 base pairs. Now, what is the beauty of this separation between these conserved sequences? That whenever there is 23 base pairs, it is going to ensure 2 turns of your DNA helix. So, if you know that there is multiple turns in the DNA helix, right? If we have 23 base pairs separating some two sequence, it is going to be responsible that we have, so we have one helix here, one helix here, right? So if it's somewhere that you have your heptamer sequence here, so we are visualizing everything like a linear structure, but essentially all of this is arranged onto the DNA coiled structure that we usually see. So if you have a heptamer here, this is present 2 turns. So this is one turn and this is the second turn. So we have 2 turns of DNA if we have the heptamer and nonamer sequence apart by 23 base pairs, and if we have this base pair difference is 12. Sorry. So we have the heptamer and nonamer sequence one turn apart if it is separated by 12 base pairs, right? So this is just something that forms the basis of your molecular symmetry, right? Now, whenever we have two-turn RSS sequence. So the two-turn sequence can, the recombination can either occur between a one-turn sequence or a two-turn sequence. It is never going to be like the recombination is happening between two one-turn or two two-turn sequences. So what does this mean? Okay. Over here, if you see, have a look at this. So you have a it's an example of kappa light chain. In the kappa light chain, we have the variable region, downstream of which we have a one-turn sequence, one-turn RSS sequence, which means heptamer nonamer sequence separated by 12 base pairs, right? That is a one-turn RSS sequence. In between the V and J. So until this point, we were calling everything that is between V and J as a intron sequence. So it is going to be spliced off. But the introns essentially are non-coding sequence, but they have a very important role to play in overall process of splicing. So overall transcript that forms, the introns do have a role to play. Understand that as something that ensures flexibility in the processed molecule that forms. Based on whatever is the nature of your splicing, you can have different combinations. That is what we were reading so far about, right? This one turn sequence, and we have a two turn sequence here. Only if it would have been, so in lambda chain, you see that this is a one turn sequence in upstream of lambda chain. In this C chain that might be somewhere here, it will have a one turn sequence. What is this one turn and two turn sequence doing? Since recombination can only occur between one turn and two turn sequence, this one turn will never go and join to the one turn sequence of your C region, and that is how the cells are ensuring that we always have a V joining a J. Because if there would be, if such sort of arrangement in present in the introns would not be there, the recombination would have occurred very randomly. So presence of this one turn or two turn RSS sequence is a way by which the cells ensure correct joining of gene segments, so that only a V segment joins to a J segment, and this V do not directly join to, say, the C segment in your light chains. Okay. And in heavy chain, definitely the question is a little more complicated because four such gene segment joining has to occur, right? So all of this is being ensured by this one small phenomena, that recombination only occurs between a one turn and two turn sequence. Okay, this ensures correctness of recombination. So the kappa light chain is arranged in a way where this one turn sequence is downstream of the V kappa, the variable kappa. So we say that there are 20 such variable regions. All the 20 variable regions are going to be followed by a one turn RSS sequence. Either one of these one turn RSS sequence has a chance to bind to either of the two turn sequence, which is upstream of all the different J sequence. Is this part clear? If there's any doubt, please stop me here. So all the different V regions are by default having a one turn sequence of a one turn RSS sequence downstream of it, and all the different J regions are going to have a two turn RSS sequence upstream of it, so that we are saying that any of the different V regions can join with any of the J region, but for sure it's a V joining the J, because only a one turn sequence and two turn sequence can participate in this recombination process, right? It doesn't matter. So in lambda chain, if you see, we have a two-turn RSS sequence following the variable lambda region, and a one-turn RSS region preceding the J lambda region. But that is completely okay. It only has to the cells only want to ensure V joining a lambda, it is not really going to make any difference if we have one turn RSS sequence in with the J or the two turn RSS sequence with the J. The only idea is to have either of these two facing towards each other so that the recombination between them can occur. Now, so a very smart arrangement. So how this arrangement is really helpful is further understood by the gene arrangement of heavy chain, where we have this D region in between two one-turn RSS sequence. Right? Very smart because it is having a two-turn RSS sequence upstream of J and a two-turn RSS sequence downstream of V. D is flanked by on both the sides by one turn RSS sequence. So the two can never go and combine here. This two only can combine here, and this one can only combine here. So first to happen is this one turn RSS sequence coming and joining here, because D and J combination happens first, and next to follow is this two turn RS, because once that recombination happens, what we have is a DH, because now these two are, so a cut is produced here and here. These two segments are going to recombine. So ultimately what we are left with is this combination, but this is still flanked by a one turn RSS. So now the attack will be from this position to this position, and the splicing is going to happen from this to this position. So ultimately we have a VH, DH and JH. Is this clear? So the idea is to also appreciate how beautifully all such things are arranged, and just try and imagine how scientists would have discovered such precise details of the body, right? So the smartest engineer is your body, which knows where to place what sequence and everything that has to come up. And next to appreciate is how beautifully people have actually decoded all these processes and mechanisms. So it's never like you have one person working. So it might be that someone identified that there is something called as RSS flanking such gene segments, and someone else showed the significance of these. So that is what scientists across the world do. They try to solve bits, a huge puzzle in bits, and ultimately you have the overall picture that comes up, and which unfortunately we have to memorize if we want to score in a particular subject. Let's just Now this is a mechanism by which, should we stop here? Should we take this up in the next class? Because I think this is, so what we understood here is going to overall form the basis of recombination that is happening. What is happening, what if you understand the concept by which this is happening, because of one turn sequence and two turn sequence, we have already talked that there is something called as RAG enzymes, and we have DNA repair enzymes participating in this overall recombination. To understand this picture of so this is essentially just the chemical reactions that is happening, which is bringing about the joining of these segments. If the three of you who are still with me can just quickly say me a yes, or we'll take this up in the next class. If you still think you have more space in your mind to accommodate this, or I think we'll just take it in the next class, because this we will start with this, so that is also going to help you with a quick revision of whatever we have discussed in today's class. Okay, we'll stop here. Please tell me if there's something else that we want to discuss further. And let me just also bring this up, because we have already talked a lot about class switching that is happening, and we will take another 5 minutes to just maybe complete with this. So, as I was saying, this class switching that is happening is also being guided by the T cells that are interacting with the B cells, right? So, how is this T and B cells interacting? We definitely have the CD40, CD40L molecules that are going to interact. And based on this co-stimulation that the B and T cells is giving to each other, we have the T cells producing certain cytokines, right? So which cytokine is being produced, that is going to guide the differentiation of the centroblast into, sorry, that is going to guide the differentiation of your centroblast into different types of. Hmm. So if we have IL-2, IL-4, and IL-5 present, we can have the proliferating centrocytes uh, from centroblast to centrocytes that can now differentiate into. So division is being triggered in presence of IL-2, IL-4, and IL-5. Once these cytokines are there in its microenvironment, this centroblast divides into centrocytes, which are now further acted upon by the presence of certain other cytokines. So if we still have the same milieu of IL-2, 4, and 5, the stimulus is in the direction of producing IgM secreting plasma cells, or IgM expressing memory cells, right? If we have IL-4 in the microenvironment, the plasma cell form now is of IgE or Ig1 type. Similarly, if we have TGF beta, we have IgA or Ig2b. If we have interferons gamma, we have Ig2A and Ig3 secretion, secreting plasma cells into differentiation. So proliferation stimulating cytokines is IL-2, 4, and 5, and differentiation initiating cytokines is IL-2, 4, 5, because it is also it also specifies that the IgM make is going to be there for the isotype of antibody produced, or it could be of interferon gamma or TGF beta. Just we will also come to class switching as we follow up with our discussion, but I think it would be just good to know that we were saying that the body knows which cell. How the body knows is because of the cytokine that is being produced by the interaction of B cell and T cells together. Okay, all of this happening in the germinal center in the peripheral lymphoid tissues. Okay, I think with this we can stop. If there's anything to ask from whatever we have understood today, please ask.
Mom, these RSS are a part of introns, right?
Yes.
Mom, these have to do nothing with the turns or the coiling of the DNA.
The RSS. So, see, when we talk about this mechanism, so RSS is a site for which is being recognized by the RAG1 enzyme. So RAG1 enzyme, we will come to it. So the recognition of RSS is happening by the RAG1 and 2. So first we have this RSS which is present near in between the V and J segments. The RAG enzyme comes and binds to this RSS, RAG1. This RAG1 is going to cleave some strand. Then we have RAG2 binding to it, and that is going to form a hairpin structure. And ultimately, two cleaves is going to happen so that we have the intron splice off. Because if you just imagine a linear structure, if you want this segment to join to this segment, one cut has to happen here and another cut must happen here so that these two ends can come together, right? So RAG1 and RAG2 is going to mediate it, but RSS sequence is going to help the correct positioning of RAG1 and RAG2 enzyme. We'll discuss that.
Okay, ma'am. And you have used the term recombination. So in the immunology, it simply means that the another combination of the genes are taking place in order to form the immunoglobulin, right?
Right. But
From the
in what
original,
stage? The germline.
ma'am, what is the location of this germ line?
See, hematopoietic stem cells, right? So whenever we have stem cells, first thing is that it can differentiate into any types, right? The bone marrow is where the hematopoietic stem cells are harbored. So we have so stem cells have the ability to differentiate into all the possible types. How so? Because it is not committed to any particular type, right? It has no particular gene expression that guides its differentiation. Based on whatever is the stimulus in the environment, you can have a stem cell differentiating into either lineages. Such stem cells are so I don't think it's scientifically correct for me to say that they are as good as germ cells, but since they are having all the genes present in it already and it is not committed, it can switch to any of the lineages. So that's how it's somewhat like the germ cell. I don't know if that's a correct
I'm not
analogy.
clear with the germ line. This term you have
Mhm.
used many times in today's class. So what I don't know anything about the germ line. So is there anything that to remember or give importance to it?
See, germ line is how do I explain this to you? So, germline, the word germline comes from the fact that we have meiotic cells, right? In so when we are talking about the sperms or the ovomes that is forming inside your, so you would know that all the cells and everything that is inside your reproductive cells there. So they have, so they are in a way similar because they also have all the cells present in it. So I think germline is a term that comes from there. uh, I'll find out a proper way of explaining this to you before I make a comment, right? I think by the next class we'll get back to it. So germline is something that has the ability to differentiate into whatever it wants to. It's something like that, but even I am not very sure of how to explain this to you. So give me time for the next class. I'll get back with it, with a proper explanation.
Okay, ma'am.
Okay. Okay. Anything else from anyone? So might be the next Tuesday's class will be shifted to some other day in the week. That depends on the availability of NPTL slot. But if possible on Tuesday, we'll do it on Tuesday, or I might just postpone it to Friday in the coming week. So you'll get a mail accordingly, right? Okay, we'll end today's session. I hope you will be able to understand the topics better and just go back and try to. So that's what I was saying. The last two class, this class and the following two classes are going to be very related to each other. It would be very good if you all can revise everything before coming from the for the next class, or otherwise you'll also try to summarize everything. We try to do that before starting every new class, right? Okay, okay. I'll see you all in the next week. Good night.
Thank you so much, ma'am.
Okay.
Thank you, madam.
Thank you.